Great. Well, thanks very much for the introduction, and the moment of levity to begin was appreciated as well. And I'm really excited to be here and be a part of this symposium, this roundtable. I've learned a lot from the excellent presentations this morning. So my presentation will be a little bit different focus than this morning, and talking more specifically about this search for pharmaceuticals from the sea, and talk about some of the successes we've had and some of the challenges I see as we go forward. And because I'm going to try and do several things, I decided I'd go ahead and put it in an outline so you have a sense of where I'm going. I just want to start with a little introduction on why do we need to find new pharmaceuticals. So I'll be rather brief in my comments there. And then, are the oceans, have they been a productive source of new medicines? That's something I'll address as well. And then I thought I would give a little bit of flavor of the research by talking through a couple of examples from my laboratory. So there's actually some chemistry in today's presentation. And then return to this issue of, will environmental stressors impact and change this field of drug discovery from the sea in positive or negative ways? And I think the jury's out. Is our knowledge, to get to the punchline, our knowledge is fragmentary, and we just really don't know, except we can say about the disappearance of biodiversity, and that will certainly be of negative impact. Okay, so we have a number of diseases that are either ineffectively treated by current medications like cancer, although we see reductions in women in lung and bronchial cancer, breast and colorectal cancers, and stomach cancer early on, and uterine cancer from various modifications to behavior like smoking or refrigeration of foods or early screening. There are other cancers that are increasing actually over time, and currently both in men and women it's about a chance of 1 in 4 that you will die from one of these diseases we know as cancer. But there's many other types of diseases that are poorly treated, such as microbial infections. We heard a little bit about that this morning, but here's another example of MRSA, which has gone from being a highly sensitive strain through several waves to become an increasingly drug-resistant strain that's now out in the community as well as in hospitals, and is a real health danger to our populations. And none of our current pharmaceuticals really work effectively to some of these multiply drug-resistant MRSA strains. So we need new pharmaceuticals to treat these types of resistant organisms, as well as new methods for their application, because we're just going to run into the same problem over and over again if we continue to use antibiotics the way we have in the past. And newly emergent diseases, and we heard some about that in some of the presentations this morning as well. That is particularly viral diseases jumping from wild animal populations into human populations and giving rise to various diseases such as AIDS and many others. And as we erode some of those natural habitats on land and have more and more contact with wild animals, it's predicted we'll see more and more viral diseases making this jump. So we have need for pharmaceuticals here as well. So where have we gotten our pharmaceuticals to date? The wonderful work of Dave Newman and Gordon Craig here at the NCI to track back for some 1,355 pharmaceuticals in the clinic, or recently in the clinic, some of these come and go, where they actually come from. And so this pie chart illustrates this, that some, well, that's about 26% in combination are natural products and natural product derivatives. So natural products has played a very important role directly in providing us with our useful medicines, particularly in areas such as antibiotics and anti-cancer agents. A growing part of the pie, though, are biologics and vaccines. So these are macromolecules, proteins, some carbohydrates in there that are used. And then about half of the pie, though, are synthetic drugs. And this is where Craig and Newman really could delve in with their deep knowledge of the origin of these drugs and ask that question, where did the synthetic chemists get the idea for that molecule? And they found that a substantial proportion, all of these parts of the pie, are where a synthetic chemist has looked to nature, seen a pharmacophore in a molecule, a particular element of it or an aspect of it, and embedded it in another molecule that's of synthetic origin that's now decorated in ways that it's orally bioavailable and it has the proper pharmaceutical properties. So indeed, about 70% of all of our pharmaceuticals are in fact in some sense natural products, natural product derivatives, or inspired by natural products. So how has the marine environment done in this regard? The unique life forms with their myriad colors and shapes and sizes and adaptations to underwater life, they have been a tremendous resource of novel chemistries. Have those translated into new medicines? Yes, they have. Here are some 9 pharmaceuticals in the clinic today that derive, in some sense, and I'll go over it very briefly, from marine creatures. These are a series of purine nucleosides that derive actually from some work some years ago with sponges and finding that they had an unusual sugar that they incorporated. And this has given rise to 3 very useful anti-cancer agents and 1 antiviral agent. This complicated curlicue is actually a representation of a peptide that's cyclized to itself to make a knot-like structure, and it's actually used by the cone snail to prey on fish and immediately anesthetize or paralyze them so that it can, if that fish swims 1 foot more, it's lost to the mollusk oftentimes. So this is like an immediate it and other peptides that it injects. This one, though, has found clinical utility as for treatment of pain, particularly chronic pain that's not responsive to opioids any longer. We have a couple of complicated molecules here. This one coming from a tunicate from mangroves, first found down in Puerto Rico, and now is sold as the natural product directly from a Spanish company, Pharmamar. And this one here is actually also from another sponge. It's actually just a part of the molecule, and that very complicated structure is produced totally by synthesis now by a laboratory up outside of Boston, Assay Pharmaceuticals, and they're one of the leaders, actually I would say, continuing leaders in trying to look to nature for inspirational new drug molecules. And these both have entered the market rather recently and are showing some good efficacy in treating various cancer types. This one here I rather like because this part of the molecule is actually called, it's a relative of dolastatin. It's a toxin that was found in a sea hare, ultimately traced back to its diet of cyanobacteria. And so this is a modified dolastatin 10 compound that's linked through this linker to a monoclonal antibody and delivers this very toxic payload directly to cancer cells. And it's showing very good efficacy in some lymphomas and other specialized cell types. And then we have some modified omega-3 fats from fish oil, modified in that they're ethyl esters, and that seems to show some improved efficacy in treating hyperlipidemic situations. So some perspective on this. Has this been very effective? These 9 drugs that only, I can count them on my 2 hands. Is that very good? It's actually very good if you look at the records out there. The NCI estimates it takes about 15,000 compounds to be evaluated in some level of biology in order to find one drug that goes the full distance. It's a very, very challenging enterprise. The marine environment has yielded these 9 compounds. MarineLit is a commercial database that keeps track of the marine natural products that are published. There's some 22,000 compounds known. And so that is a success record of 1 in about 2,500 compounds. So that's about 6-fold better than what the record has been from other sources. That might be synthetic libraries or other traditional land-based sources. So now I'm going to segue into some of work in my lab to give you a little bit of a flavor of how this occurs. And I actually work with cyanobacteria, and they've come up in a negative light earlier this morning. And so here we'll see a positive spin on this, I hope. They're ancient organisms. They have a wide range of genomic sizes. On the larger end, they have a lot of genetic capacity to hold these complicated genes that code for the enzymes that make these exotic molecules. And hence, they're very, very rich in their bioactive compounds. So we go out in the field, as Jack indicated, we do get some field time, and we collect samples for chemistry, but we also preserve the organisms for their DNA, because increasingly this field is moving into the genetics of natural products biosynthesis. We extract the materials and then test them broadly for biological activities, and I have a lot of collaborations in private sector, in government, in other universities, and internationally. And I think, if the chemistry is broad, biological space is even broader, and that's something I think we could do even more of. But once we find a hit is then to use that biological assay to guide the isolation of the compound and define its molecular structure, and that becomes the lead for lots of other areas of investigation, such as how does this molecule work? Sometimes the lead is most valuable in that it reveals some new way to kill cancer cells. And while that lead is not going to go into the clinic, some other molecule perhaps will. Can we synthesize the molecule and make analogs to really come to understand how these molecules work at their targets? And can we understand that biosynthetic process, maybe even capture that biosynthetic process in some way? This is at the frontier of this field. Why are these molecules made in the first place? What is their chemical ecology? And then there's much work that goes into trying to develop these into actually useful agents. So I love this map of vascular plant biodiversity. I wish I had it for the marine environment, but the point is around the tropic belt, both on land and sea, is areas of highest biodiversity on our planet. And so we have some biodiversity hotspots in the Philippines and Indonesia, and then down here in Panama. And it explains in part why usually in the wintertime I go to the tropics and to the ire of my dean. No. But we do go to areas of high biodiversity where there's lots of plants and animals all vying for that same rock to live on, to harvest sunlight, to access nutrients. And it's that very competitive nature of that environment that seems to drive the production of so many very biologically active molecules. And so we'll go back into mangroves, which are a little icky to swim around, but these branches and roots are actually very nice substrates for cyanobacteria and many other organisms like the tunicate that gave rise to that drug actinocidin that I mentioned earlier. We also go collecting out on reefs and collect some of these cyanobacteria into just little simple Ziploc bags, bring them back into the laboratory, and we can grow them if we've brought them back in a living state. And two stories I'm going to tell, again, very brief format, but one comes from our work in Curacao. Actually, my students were there without me for this, and one of my students came down with dengue fever, and he was sick for 6 months with that and seriously ill. I've had dengue once as well, but was not sick for that length of time. So the other student, of course, can't go scuba diving by herself, but she did go out snorkeling, and she found just on a mooring rope the cyanobacterial growths. It turns out that that's been just an amazing discovery for our laboratory. Again, very short form, we used a cancer cell line assay to guide the isolation of a couple of molecules. It was assisted by some NMR analysis. And we came up using the standard techniques that we use and to come up with these structures, which were quite novel in the occurrence of this epoxide, that three-membered ring next to that ketone, that carbon-oxygen bond. There's a class of drug that's just been entered into the market that has exactly that same configuration of epoxy ketones. They're very, very powerful anti-cancer drugs. So we came up with a chemical synthesis for this compound, and I don't want to go through the synthetic chemistry, but the point is to say that we were able then to use that chemical synthesis to make about 12 additional analog structures, and we're continuing on that process. And this has been very insightful as we now have these various molecules that we can see some are very high potency at killing cancer cells and some are inactive, and we start to learn structure-activity relationships in this drug class. And that's taken us in wholly other areas of science, such as we've crystallized one of these molecules with this enone into the active site of an enzyme and done X-ray crystallography of that to really understand exactly how it binds. It turns out this particular compound binds in a way unlike any of the other drugs in this class. And so it's revealing new ways to interact with the human proteasome, which is a very effective drug target. So the human proteasome, it's the machine, it's a series of, I think there's 29 proteins in all that form a little barrel that acts as the way to degrade all of the proteins that are turned over in our cells. And it's a very good target for anti-cancer therapy, but it's a target for many other therapies as well, because the proteasome's involved in all these processes. One that's captured our interest a lot is in neurological disorders, actually with a colleague of mine, Tom Murray, out in Creighton University, where he screened our analogs because of the involvement of the proteasome for neurological properties and found that at very low concentrations, 10 to the minus 10 molar, this is like a drop in a swimming pool size, to give some sense of that concentration, causes these neurons to develop these neurite projections, and they become highly branched. And here's the dose responsiveness. In other words, we haven't really found the lowest concentration yet. So we're very, very excited about this molecule because this could offer a therapy for various conditions such as stroke and regeneration of neurons or spinal cord injury. So I think that this molecular class has a lot of future in both cancer and in the neurosciences. A second very short story will come from our work in Papua New Guinea. We've made a number of expeditions there over the years. It's one of the most dramatic places in the world to dive. A lot of these very sheer underwater cliffs. That's actually my daughter. She participated on one of these expeditions. And as you turn your attention away from out here where there's usually sharks swimming back and forth, though they're well fed, so we've never had any problems there. But, and look the other direction, against the reef, you find covering some holes, sort of a cotton-like mass of cyanobacteria that grows as a cover. And as you go to collect that, this shrimp comes out with this one large claw, and underwater you audibly hear that snapping sound of that claw. And if it catches you, you're going to get one of my students did get a little nick on his finger. And so, but it's voraciously defending its home because it weaves this cyanobacterial filaments into place, and they're attached to the reef to form a cryptic covering over that hole where it lives. But it's also defended under that cryptic covering, and nothing eats it because this cyanobacteria makes this very exotic-looking cyclic molecule, and it has some very, very pronounced biological activities, specifically anti-cancer activity. We've worked on it along with a group in Hawaii. And we found that with my colleague Fred Valeriot in Detroit that it has some really exceptional cytotoxic activities. But Fred does some very careful work. There's a million ways to fail in this and only a few paths forward. But Fred, by being very meticulous in his approach, found that the original approach of applying the drug to the animals was you could predict would be ineffective. So he applied it with a different schedule for drug administration and found that it had very good in vivo anti-cancer effects. So it is continuing as a drug lead in our lab in collaboration with a pharmaceutical company. So we've become very interested in this exotic molecule, how this is made, how this little chicken foot-like structure, it's called a T-butyl group. And it's really unique in natural products. You don't see it very often, and so we wondered and puzzled how that's made. And we thought that our prediction was that it was maybe these 2-carbon acetate units that assembled, and then there's a decoration with methyl groups from something in our cells that's sort of the universal methyl group donor called S-adenosyl methionine. So we wanted to study this further, and to do that, we had cultures of the cyanobacterium growing in the laboratory in pans about that size. So indeed, they don't grow as a veil like they do in nature. It's really the shrimp that, weaves this into that mat. And under the microscope, though, our cultures were contaminated. Here's the cyanobacterial filament under the microscope showing fluorescently sort of orange color, and we've stained it with something called DAPI, which stains the DNA of the nucleus. And we see there's lots of bacterial cells on the exterior sheath-like material of this filament. And so we had a so-called metagenome. We didn't have one organism, we had many organisms. So we had an idea, and it came from some fantastic work of one of my students. He did this scanning electron micrograph where you can now see this filament receding into this sheath. It's like a straw with these series of coin-shaped cells that go back into this sheath material. We had the idea here on the exterior, it's very dirty with lots of bacterial cells. The interior is absolutely clean, essentially axenic, free of any bacteria. Could we reach in there and pluck out a few individual cells and in that way get just the pure DNA of the cyanobacterium? Well, no, we didn't do it quite that way. I'd like to believe that we did, but I won't show this whole film, or I won't show any of it actually. It's a very finely drawn pipette micromanipulator where we were able to actually squeeze out some cells and then suck them up and then pass them through a series of wash stages to get individual cells. And just this is one of the exciting things about being in this field at this time is the access to new technologies. We can take single cells now and take the DNA of a single cell and amplify that to produce micrograms, femtograms of DNA to micrograms of DNA, and then do DNA sequencing to get the genome sequence of this organism from a single cell. So this was done at UCSD with some new computer algorithms. We don't get quite all of the DNA, but we get a lot of it. We estimate the genome size to be about 7 megabase, and we got 6.5 of it. But we found some contigs that would code for the genes that would code for the enzymes that would make this molecule, and we could recognize them because of the predicted biochemistry. And so we sequenced that, and we had to do a number of other things, but I won't go into it here, to come up with the entire biosynthetic gene cluster. And this is a process. It's a Henry Ford of the cell, in that it starts over here, and it just progressively builds this molecule, passing it down, attached to the enzyme all along to make this molecule in an assembly line type process. And so this has captured the imagination of many, that this might be a way of harnessing the power of the cell or the pathway that we could engineer, take out a piece, stick in another piece, and make new molecules at will. So as I said, this is at the frontiers of this area. Okay, I'm going to change gears to my last part of my talk here and address this issue of can I say anything about environmental stressors and their impact on our ability to discover new pharmaceuticals from the sea. And issues or stressors that came to mind as I prepared my talk was a couple of things from overfishing or fishing apex predators, which has had a remarkable effect on some islands in the South Pacific I'll share with you. Nutrient inputs certainly have a big impact on species composition, and climate change and ocean acidification issues. And so just two examples that I'd like to give from this, the first being from the Line Islands, starting up in the north of Kingman Reef, which is uninhabited. It's just reef, but Palmyra Atoll, which has a resident population of about 6, and then scientists can go and spend a couple of weeks to a month there during a summer season that it's open. It's a research station. So essentially uninhabited now, although it was hugely inhabited in World War II with much destruction to the local ecology as they put in landing fields and the likes. But it hasn't been inhabited to any extent for many years. And then progressively becoming more and more inhabited till we get to Christmas Island in the south. And what you see up in the north is most of the biomass that's on the reef are large fish, big snappers, sharks, turtles, dolphins, melon-headed whales, all and in coast to shore. And this was a big surprise, is to find that the majority of the biomass in these uninhabited reefs is in the large predators, and that the small, we tend to think of a pyramid of a lot of algae growing to some herbivores, to a few predaceous fish, and a few apex predators. What they found was an inverted pyramid. In terms of standing biomass, there's very little algae. There's a few herbivorous fish, a few more of the small carnivores, and most of the biomass at any given point in time is resident in these large, predaceous species. So what one sees is a progressive removal of these large fish as one goes down to Christmas Island, and you have substantial inputs of nutrients into from human populations at Christmas Island. But this correlates directly with increasing cover of algae, decreasing coral cover, ultimately to very degraded environments at Christmas Island. Degraded and supporting a rich complement of cyanobacteria, which is, I've just been telling you, has been the source of inspirational molecules that we've been studying. So there's some irony, there's some complexity. It's not, as we've seen in many of the other things we've talked about today, it's not necessarily a single story and absolutely straightforward. But I think we'd all like to rather see the environment looking like this. And those cyanobacteria are up there. They just don't grow in the large dominant populations that we see in the southern parts. Okay, a second short story I'd like to share with you is coming from some work with a summer student this last summer, actually. Alexis Brown came to UCSD from University of Maryland, Baltimore County for, it was in a program, I think she was with us about 2 months. And getting to know her and talking about things, she was interested in natural products and climate change. And we kind of hit on this as an area that would be interesting to study. Would ocean acidification impact the production of secondary metabolites in some of the species that we work with as a representative maybe of others, namely cyanobacteria. So we hypothesized that ocean acidification might actually have some impacts both on growth as well as toxicity and the elaboration of these natural products by a strain that we've studied quite a bit of, so we know the chemistry quite well. In fact, we've sequenced the genome of this organism and we renamed it as a result of that. So with this organism that is sort of a model in our laboratory, we set about and we thought that such alterations could impact, of course, drug discovery efforts. So we set up some 9 pH regimes, increments of 0.1 pH units through 8.3 above historical records of ocean pH down to, hopefully, we never get to 7.5 with 3 biological replicates. So but I would say this is preliminary. It's not published yet, but I thought it was intriguing. And we let it grow out for a couple of weeks. These are slow-growing filamentous cyanobacteria. And then we tried to take a number of measures of wet weight, extract weight, and metabolite content by liquid chromatography, mass spectrometry, using some standard curves. And what we found, actually not so surprising, as we go to more acidic pHs, we see a destruction of the cellular structures. Here under the microscope, you see those coin-shaped cells inside this sheath. Here we can see some destruction of that. It might be a little hard to see in this light, but there is. And then ultimately at 7.5, these were not very viable filaments at all. So we saw a concordant decrease in biomass and extract weight with decreasing pH. But we also saw a very substantial decrease in the production of these compounds. So the numbers are small. We're going in each case from 7.5 up to 8.3 for these 3 different compounds. Gerwick A is a molecule we discovered from this organism, has powerful anti-cancer activities. It's an anti-tubulin agent and has been studied by the National Cancer Institute over a number of years. Barbamide, all 3 of these compounds we discovered. But you can see the trend is there. The data are not super clean. It's hard to actually maintain the pH of seawater without some very specialized gear, which I'd like to do, but we didn't have the gear for this summer. But there is very definitely a trend from low pH to high pH that we have this decreasing amount of, per weight basis, decreased expression of these natural product pathways. So the conclusion, and again preliminary, is that ocean acidification may decrease not only the growth of this organism, but also its ability to produce these secondary metabolites, which are important to its ecology. And so we may see some decreased populations of cyanobacteria even in places where they normally grow, not subject to human involvement. So some final thoughts is both historically and currently, nature is the best source of inspiration for the discovery of new natural products. We've had some real problems with the pharmaceutical pipeline because of some turning away from this in favor of other approaches. Those other approaches haven't worked out, and there is a turn back to looking in different ways at natural products for this type of inspiration. Integrating methods from various fields of science, as some I've tried to communicate that some here today, I think is really the future of this field, and has been why it's retained so much, I think, vigor, is that it is a very multidisciplinary group that's involved in this type of work. Obviously, marine environment's changing. The coral coverage is dramatically decreased. It was recently surveyed historically over, I think, about the last 40 years. And here's a point that I would really like to make, is I think we have only a fragmentary knowledge of these more subtle impacts. Okay, maybe it doesn't change observables of this organism from ocean acidification. Namely, maybe its size, or maybe it's not subject to calcium carbonate deposition, so it doesn't suffer in that way. But what about these more subtle gene transcriptional events? We don't have knowledge of that. We barely know the pathways and the molecules involved. So I think this is an area that could use some further attention if we want to really get into the nuts and bolts of it. And so my final comment is that our health through these natural products is intimately tied to ocean health. So with that, I look forward to our discussion. Thank you.